Hello, I'm Sein Kang,
And I'm Jimin Jung.
and we are from Incheon, Korea. We conducted research on the effect of bowel function of the large intestine on the intestinal immune cell activity in mice. The composition of human microbiota, or intestinal microbes, depends not only by genetic factors, but also by environmental factors such as antibiotic overdose, dietary habits, and stress. Changes in these factors induce dysbiosis of the intestinal microbes, which are closely related to autoimmune diseases, obesity, diabetes, and infectious diseases. Also, environmental issues are deeply related into these factors. Recent studies found out that exposure to air pollution affects on gut microbiota. Also, microbiomes are closely related to environmental issues in that technology to decompose environmental pollutants using intestinal microbes are being developing. I hope our research contributes to improving the understanding of intestinal microbes and solving environmental problems. Analyze the changes in the distribution of intestinal microbes and the secretion of cytokines from immune cells when ingesting constipation-inducing substances, which is tannin and gluten, and cellulose, which is abundant in dietary fiber that promotes bowel movement. Prior to explaining our research method and procedure, I would like to tell that our animal testing was reviewed by the In-School Research Ethics Committee. First, I'd like to introduce our mouse breeding environment and food program that has been going on for 5 months. For the study, three experimental groups, tannin, gluten, cellulose groups, and a control group, were set. The experimental groups were fed a mix of normal food and each substance in the ratio of 10 to 1. The weight gain was measured every 7 days to calculate the weight gain ratio according to the food intake. The environment's temperature was 22 to 26 degrees, and we maintained appropriate humidity by the cage program. We fed an average of 400 grams per week, and we washed and changed the bedding every 2 times a week, which was a very hard work. Experimental and control feces were sampled twice, and after 1 month and 5 months from the start of the experiment for metagenome sequencing and analysis on distribution of beneficial and harmful bacteria. After 5 months, we prepared inflammatory agent DSS in a 5 weight percent concentration and supplied it as drink for 1 week instead of water for all four groups. Through the week, clinical indicators were measured and scored. Then, after euthanasia, we did three main things for post-processing analysis. The first analysis is metagenome sequencing analysis, and the second is analysis of the scored indicators, and finally, immunity change analysis. For immunity change analysis, multiple cytokine concentrations in the serum obtained through mouse blood collection was measured through ELISA. Let me explain the results of metagenome sequencing. As the RDP analysis results, the tannin group showed similar species composition compared to the control group. However, the gluten and cellulose group showed different diversity patterns, prominently at the family and genus levels. Based on the heat map analysis, the control group showed similar species diversity of the intestinal microbes with the tannin group, and the gluten and cellulose groups showed similar diversity to each other. This table shows the results of alpha diversity analysis through metagenome sequencing. In terms of species diversity, the most significant difference compared to the control group was measured by the cellulose group, which with the Shannon index of 6.53. The ratio of beneficial bacteria in the RDP index was lower than the tannin group and significantly increased in the gluten group compared to the control group. The ratios of harmful bacteria were similar in all groups fed with tannin, gluten, and cellulose. This table shows the study results of butyric acid-producing bacteria's distribution radius, known to activate mucosal immune tissues among intestinal microbes. In the NCBI index, the radio of butyric acid producing bacteria was relatively lower in the tannin and gluten groups compared to the other groups. In the RDP index, the radio was lower in the tannin group. Both the control and experimental groups showed weight loss by DSS treatment. The weight loss was highest in the tannin group and the lowest in the cellulose group. Motility was measured by assigning a score based on hunching, caused by changes in mouse movement and physiological problems in the body. Tannin and gluten groups showed a tendency to decrease motility in a similar pattern, but the Mann-Whitney test showed no significant change statistically. The cellulose group showed almost no changes in motility, similar to the control group. When the mouse's health deteriorates, the coat condition is negatively affected as the original brown turns yellowish. Criteria were determined based on the change in hair color and shape of the mouse coat to score and measure the change in the mouse coat condition and pillow erection. Tannin and gluten groups showed a tendency to decrease coat condition and pillow erection in a similar pattern. In the cellulose group, almost no change in coat condition and pillow erection was observed compared to the control group. Criteria were determined based on the perianal cleanliness and changes in mouse feces to score and measure the two factors. Mucous feces and fecal blood were observed in all three groups. The tannin, gluten, and cellulose groups showed similar levels of perianal cleanliness and changes in the mouse feces, but the Mann-Whitney test showed no significant changes. Analyzing the RGB values of the fecal sample images to judge the fecal state showed color changes in the experimental group, but except for the controls. After DSS treatment, ulcers developed due to the colonic surface inflammation induced by inflammation-related immune cells. Scores were assigned based on mouse colonic ulceration, and the mouse colonic surface inflammation was compared and analyzed before and after DSS treatment. The colonic surface condition was significantly changed in both the control and 3 experimental groups after DSS treatment. The results show that all of them had a significant inflammatory reaction in the large intestine.
Next up, by measuring mouse colonic surface inflammation, every group showed significant inflammatory reactions. The most severe cases were tannin group compared to the control group. Now, after measuring blood cytokine concentration after DSS treatment, interleukin-6 and TNF-alpha increased significantly in all groups. By measuring control and experiment group cytokine concentration after DSS treatment, interleukin-6 concentration was higher but not significant in tannin and gluten group. On contrast, cellulose group was significantly lower. And interferon leukin showed no relationship between inflammatory reaction. These results confirmed inflammatory agent DSS caused increase of interleukin-6 and TNF-alpha concentration and reduced concentration of immunosuppressive cytokine interleukin-10 to active inflammatory response. Now, after we measured the mouse colony immunofluorescence staining analysis, in both situations of before and after DSS treatment, immune cell, T helper cell, T regulatory cell, T cytotoxic cell, and plasma cell existed in lamina propria. Now, comparative area analysis of Alexa Fluor 488 and 647 showed that T helper cell and plasma cell activity increased after DSS treatment. Analysis through ImageJ programs for these types of T cell showed that cytotoxic T cell activity decreased compared to the control groups in all experimental groups, especially in gluten group. For helper T cell and regulatory T cell, activity of both cells increased in cellulose group and decreased in gluten group. Finally, based on this data, the gluten group showed positive side to intestinal microbiota composition, such as increased species diversity, and beneficial bacteria distribution. However, it showed significant decrease in T cell activity after DSS treatment. In tannin group, it showed negative microbiota composition by reduced species diversity and beneficial bacteria distribution before DSS treatment. Following that, it showed rapid weight and motility loss and had least inflammatory response. Cellulose group showed positive change in microbiota distribution with increased species diversity and beneficial bacteria distribution. Plus, positive changes showed in immune response with increased immune cell activity after DSS treatment. In conclusion, long-term consumption of two constipation-inducing substances resulted in changes of intestinal microorganisms and significant change in immunity resulting in immunocompromised states. In the future research that will follow, reconfirmation of whether tannins and gluten are actual reasons for constipation in the intestines is needed. In addition, it is necessary to conduct a more long-term analysis to reveal exact regulation mechanisms between diet, intestinal symbiotic microorganisms, and the immune system. Thank you for listening to our presentation.